Welcome to the Genome Center at University of California, Davis. I'm Richard Michelmore. I've been on campus for a little while, since 1982. And I've had responsibility for lettuce genomics with a particular emphasis on breeding for disease resistance and understanding how plants are resistant to a multitude of diseases at both the classic genetic level and the molecular level. During my career, there's been a tremendous revolution in our ability to identify variation and now manipulate it, as we're going to describe in this video. Plant breeding is all about the basic equation: genotype plus environment equals phenotype. So what we see is the phenotype, but that's a combination of the influence of all the genes in the genome plus the environmental effect. This is why in human twins, they won't absolutely be identical. There is always an environmental effect. So what we want to do with plant breeding is to improve the phenotype. And that requires an understanding of the genetics. But also it's in the context of the environment, or the different environments that the crop gets grown in multiple locations. Well, when I started, we had very little idea of the genetics of lettuce. We had no idea of what the genome of lettuce looked like. Now we know we have a gapless telomere to telomere, that's end-to-end DNA sequence of lettuce, which is quite remarkable. It turns out that lettuce has the same size genome as humans, about 2.7 gigabases, though it's got a few more genes than humans. So lettuce is actually more complex than humans are. So it's up to the plant breeder then to manipulate the genetic variation. So it's really important to have a resource of genetic genes that you're interested in, often of which come from wild species. Initially, you have to understand the genetic basis. And you do that really in two ways, one of which you can make crosses between two lines that are different for your trait. And that's a biparental mating. And you look at segregating progeny, and then you try and work out how many genes there are and where those genes are on the chromosome. Once you've established that, you can then develop molecular markers for that trait. So this will be markers that there are a variety of different technologies, but basically you ask at the DNA level, how can I select for that phenotypic trait? And increasingly, seed companies are using multiple molecular markers for traits of interest in many crops, of which lettuce is one. Disease resistance is one of the ones that is often simply inherited, and by using molecular markers, breeding companies can select for multiple diseases without actually having to do the disease screens, which are often complicated, and sometimes you just cannot combine different combinations of resistance genes without having the molecular markers. One of the things that's really important with disease resistance is to understand variation in the pathogen population. So not only do we study variation in the host, the plant lettuce, but we also study variation in the pathogens, because there's a continual game going on that the pathogen is trying to infect the host is trying to detect the pathogen and elicit the resistance genes. And so you get this boom and bust cycle whereby the breeder will recognize a disease-resistant line, probably in a wild species. They integrate that gene into the cultivated lettuce. If it's successful, the acreage increases of that particular cultivar or cultivars carrying that resistance gene. That puts a steep selection pressure on the pathogen to evolve to overcome that resistance gene. And this is what's called the boom and bust cycle. You go round and round and round. Now the goal of plant breeding then is to slow that cycle down. We need to maximize the evolutionary hurdle for the pathogen to overcome preferably a pyramid of resistance genes. So what you need is an understanding of the pathogen population, but you also need to have a reservoir of genes that you can then deploy. Probably, hopefully, 3, 4, 5 genes at a time. That puts a very steep selection pressure on the pathogen population. All pathogens will continue to evolve. And of course, a classic example that we can see in real time is the SARS-CoV-2 virus. It has evolved over time. You get all these new variants. And people have asked me, I did a lot of COVID screening, that was it different working on the pandemic? And I say no. I've been working on pandemics for the last 40 years. They're just plant pandemics. And actually plant pandemics are a global threat to food security. They happen all the time. And therefore the role of plant breeders is to counter this threat and hopefully stabilize global food production. But it's a continual process. Breeders cannot take their eye off the ball. Now, getting back to ways that we can identify resistance genes, one of which I said was a biparental mating. But that only captures variation in the two parents. Another way to do it is what's called a genome-wide association study. You have large numbers, hundreds of different accessions, and then you screen them, you have your phenotype, and then you look at their DNA sequence and you try to find a correlation between a particular region in the genome and your phenotype. So a GWAS, genome-wide association studies, is another way of understanding genetic variation. Now all of this builds on to analyzing variation. As you'll hear later on in the video, we now have ways of manipulating that variation through either gene stacking, which is marker-aided selection, or particularly exciting is genome editing. Plant breeding is a cycle of increasing and decreasing variation. That you have a well-adapted cultivar, but then you will make crosses on the basis of the knowledge of genetic variation we've just been talking about, and that of course increases the genetic variation, and particularly if you've made a cross to a wild species, most of that genetic variation is not desirable, and therefore you go through a selection process that decreases variation. And so we go around this cycle of increasing and decreasing variation. Now, it's a slow cycle with traditional plant breeding, maybe 10, 15 years that you go around before making the initial cross and actually having a proven cultivar. Now with marker-aided selection, that is if we have markers for the traits we're interested in, you can reduce that time maybe by half. As I say, you can also combine traits that you wouldn't be able to with traditional methods. So breeding companies now are, I don't know the exact number because it's increasing all the time, but in lettuce I believe the bigger companies are maybe doing marker-aided selection for 6, 10, 12 different genes, mainly disease resistance. And across all their crops, they may be generating over 1 million data points a day in order to do this selection. This isn't large scale, this is industrial scale marker-aided selection. And it started with the corn and soy companies, but now the vegetable companies are also doing it. The way marker-aided selection works is you can analyze the genotype of a very young seedling. You don't have to grow the whole plant up. So what happens is the breeders will grow up large numbers of seedlings, and they will take a small part of the tissue and they send that off to the lab. The lab then extracts the DNA from that sample, and it will run a whole bunch of molecular markers, and often what they are is they're PCR, a pair of PCR primers that will distinguish different versions of the gene. And then from that data you get a large spreadsheet with your genotype and all your individuals, and then on the basis of that the breeder will choose the little seedling to grow up into a big plant and probably make another cross with it. So it's really, again, it's the advantage of molecular markers is it's the efficiency, and you don't need to grow large numbers of plants to maturity. As we've just been discussing, genetic variation is really key to advancing plant breeding. We live, however, right now in unprecedented times in our ability to detect and to manipulate genetic variation. And there is not just massive opportunities for marker-aided selection, but we can also increase the genetic variation we're seeing. We can now make precise genetic edits using CRISPR-Cas. Also, there's AI. The impact of AI is, we don't really know, like in many aspects of society, but AI is going to have a fundamental impact on the way that we do plant breeding. We are faced with our ability to generate unprecedented amounts of genetic information. We can generate unprecedented amounts of phenotypic information. We have unprecedented amounts of environmental information. And it is beyond any one human to be able to analyze and put all that together. But with the aid of machine learning tools, we're going to be able to come up with novel strategies, novel ways of manipulating variation, or understanding variation and the impact of it. One of the technologies that we now have is very efficient genome sequencing and assembly. It took my lab 15 years to get a complete genome complete genome assembly of one lettuce cultivar. We can now do it in 4 or 5 weeks. And therefore, not only do we have one genome assembly, we are in the process of generating over 100 genome assemblies, representing the diversity in the gene pool available to the plant breeder. And so, when we do our genetic analysis, before we could get down to just a region of the chromosome from which we could design molecular markers, but we're getting quite close now to being able to actually get the causal gene, the gene that's responsible for that phenotype. And this is required if we're going to do genome editing. You've got to know the causal gene, not just have a molecular marker. That's really important. Now, another area that's going to be really important in the future, as I said, much of plant breeding is dealing with disease resistance, and that's been the focus of my lab. But the big challenge of responding to climate change is resource use efficiency. That is, how can we use water, nitrogen fertilizer, and phosphate most efficiently? Because that's going to be absolutely critical to maintaining yields. Now, in terms of resource use efficiency that I've just talked about, water, phosphate, and nitrogen, much of the plant is getting that through the root system. And that's an area we really don't understand very much about. The root system is very plastic, but there are clearly genetic differences between different lines, different cultivars, in the way their roots are configured. But it's very challenging to look at roots. So we can study root seedlings, we can study them in hydroponics, but soil is complex, it's large, and one of the frontiers now is to really understand root architecture and how we can breed for the optimal root architecture. Now, you can't just say we need bigger roots, because if the plant is putting all its energy into bigger roots, it's not going to put it into the above ground where you're harvesting, either be it seed, be it a head like in lettuce. So you need this optimal trade-off between the allocation between the roots and the shoots.
I'm Luis Salazar. I'm a PhD candidate at UC Davis in the MIchelmore lab, and we are at the Genome Center. So my PhD is in looking into the genetics of salt tolerance in lettuce with a specific focus on wild species. So the goal here is to kind of look at the genetic variation in lettuce, especially in wild species, to see what the genetics is like, so that we can potentially bring in alleles or genes from wild lettuce to domesticated lettuce for salt tolerance. And the reason we're doing that is to improve the salinity tolerance of lettuce in the face of increasing temperatures that cause increasing salinity. So with my PhD, there's kind of two halves. The first half is what we call forward genetics, where I'm interested in traits or a phenotype and then dive into what the genetics are behind that. So with regards to some of the technology that we use, I am using hybrid populations to look into traits related to salt tolerance, and we are able to use things like sequencing to develop markers so that we can develop genetic maps for these genetic studies. So there's multiple ways you can approach it. Definitely there's a little bit more of like the production side, trying to kind of replicate either greenhouse production or like things in soil. So part of my PhD involved doing greenhouse assays where I grew plants in a media, put them on drip with a saline solution and see how that affected the plants. The other half is doing genetic studies of particular genes of interest. So thanks to, again, where we are with biotechnology and specifically lettuce genetics and genomics, I've been able to essentially use biotechnology to manipulate genes that we believe to have known function in salt tolerance. So that involves using transgenic technology to either overexpress genes or using CRISPR-Cas9 to edit genes to knock them out and see how that affects plants, or specifically how that would affect lettuce and its salt tolerance. And that would be more kind of in what we call in vitro, so like in plates of agar or media, basically kind of more in sterile environments. But the end goal would then to be bring that out to something like a greenhouse experiment and see how that affects both kind of at a smaller scale, but then at the more at a scale closer to what we, how lettuce would be grown in the field. Basically, I take seedlings, grow them on these plates with or without salt, and then after about a week and a half, I take photographs and I trace out the roots using this program, and then use that root architecture data to then dive into the genetics and identify regions of the lettuce genome associated with these root traits.
My name is Amber Flores, and I'm a PhD student in Dr. Richard MIchelmore's lab. And we are currently in our tissue culture room where we do our plant tissue culture. Tissue culture is the process of creating a whole new plant from a single tissue. So this is the way that we can create transgenic plants that we use to study the function of genes, to create lettuce that has better qualities. And so what we do is we take the cotyledons of lettuce, and so those are the first leaves that come out of the seeds. We incubate them in a bacterial culture that has agrobacterium in it. So agrobacterium has this really cool natural function where it's able to take a portion of its own DNA and basically spit it into the plant genome. So what we do is after we incubate those cotyledons in the bacterial culture, we then put them on plates like this that have hormones on it. So there's hormones in the media that cause that baby leaf to start to divide, and it creates an undifferentiated mass of tissue. So maybe this is not the best way to think about it, but you can kind of think about it as like a little tumor. It's just like a clump of cells. And so once we have this clump of cells, which we call a callus, we can then put it on different types of plates that have different ratios of hormones that lead to things like the creation of leaves. So this one here is on a plate that has hormones that stimulate the growth of leaves. And so you take that ball of undifferentiated cells, put it on a plate, it tells the cells there to become leaves, and so then we have leaves. And then after this, after we have sufficient number of leaves, we then transfer it to another type of media, and this is a little bit harder to see, but this media has hormones in it that tell the plant to start to create roots. And so then once we have this plant, we have the shoots and the roots, we have a transformed plant. And it has whatever we engineered the agrobacterium to contain. And so this is the basic way that we make or transgenic plants to study different genes in lettuce. One common thing that we do in this lab is we create transgenics using CRISPR-Cas9. So CRISPR-Cas9 is this amazing novel way to make very precise edits to the lettuce genome. And we do this to improve lettuce. So I study carotenoid biosynthesis. So carotenoids are an important nutrient that us humans need to eat, and it has provitamin A capacity. So carotenoids are the reason why we can biosynthesize vitamin A. And lettuce, unfortunately, has a very low amount of carotenoids. So I use this process to create transgenics that contain CRISPR-Cas9 that are targeting specific genes within the carotenoid biosynthesis pathway so that we can improve the amount of carotenoids in lettuce. I grew up in Los Angeles, and I'm the first in my family to actually leave and to seek a 4-year degree in the sciences. And so when I first entered undergraduate, I was really interested on why people ate the specific foods that they ate. So I was more interested in the nutrition aspect of our food system. And so while I was here, I began to take classes that talked about agriculture and how agriculture was a very important, link in the chain of our food system. And so once I realized that, I was like, wait a minute, I can actually study something where I can improve the crops that we develop and make them more nutritious. So that's kind of where I ended up, and that's why I'm here, is because I'm really interested in using plant biology to improve the nutritional quality of our crops. If I'm being fully honest, at times when you're the first in your family to do something, it can be kind of isolating. And sometimes it can be hard to find the motivation. I think what has become really important is just to continuously remind yourself, again, what is driving you and why you're doing what you're doing. So to be able to complete a PhD, you really need to have some sort of internal motivation and reminder exactly why you want to do the research that you are trying to do. So it does take a lot of hard work, but if you build your community, you eventually find folks that are there that you can lean on and take your mind off things when things during the PhD get a little bit hard. So as far as completing a PhD, you need to have a lot of motivation, strong work ethic, and then also build a community that you can rely on when things get tough. And so that's also really important.
Looking back over my career of the past 40 years, I feel incredibly privileged that to have been through this tremendous revolution in technologies available to us. But also, it's extremely exciting looking forward that, and being a plant breeder is a remarkably rewarding career. Not only are you generating new data, people are actually interested in the data that you generate. I have a very good interaction with the seed companies. We do foundational research that's not competitive. We will generate the genomic resources, the genetic information. They then take that and they collectively fund me for that, but then they compete as plant breeding companies. And it's a really rewarding career. And I mean, I strongly encourage you to consider plant breeding because you're never going to be out of a job. And actually, you get much more valuable the more experience you have. You get to know your crop. You become really the world authority on a particular crop. And sure, it's going to be very computational, but it's going to be a very exciting next 20, 30, 40 years.